Hi everybody, it's Mrs. Peachy from your WCA biology class, and I'm going to be doing a tutorial for you today on the portfolio on fungi, which is in Unit 2, Lesson 8 of second semester biology. So this was kind of interesting because we will be using an outside website that enables you to use kind of a virtual microscope, multiple different types of microscopes, and view some images of different types of fungi under the microscope, which is kind of neat. And you'll be able to see a lot of the different characteristics that are found in fungi, along with, like, asexual reproduction and spores and all sorts of kind of neat stuff like that. So when you go to your course tree, you will be looking at the virtual lab sheet, which I will click on and open for you. And I will bring that screen over here for you here. So this again is another PDF file, and just like, all other PDF files for the school year, you are going to be making sure you right click, save as, and select a place on your computer to save it that you will make sure you'll be able to find. So I would suggest saving it to your desktop because that's probably the easiest place for you to locate it. And maybe saving it as like Fungi Lab with your name, because that makes it a little easy for you to find it when you are attaching your file later. Please make sure you do save the PDF and that you close out the one that appears in your web viewer, and then you open the file directly from your desktop before you save it again with all of your information. If you don't follow those directions, you will more than likely not save your information, and you'll submit a blank document to me, which means you'll have to redo it, and that's just a pain for everyone. So hopefully it'll work well for you. Always double check what you're going to be submitting. Make sure it has information in it before you attach it to the Dropbox. Okay, the second thing you're going to need to do is to open the actual virtual lab. So I'm going to click on the second thing here that says Virtual Biology Labs. And when you do that, that takes you to this website, Virtual Biology Labs from Brigham Young University. So the first thing it's going to ask you to do, and I just keep moving my picture around here, is it's going to ask you to select yeast from the list here. So I can kind of scroll down here till I find yeast, which is at the very bottom. And then it's going to ask you to select some thumbnail images from above the microscopes. First thing I'm going to need to do is I'm going to need to click over here where it says my microscopy lab. And I'll click on that. And that's going to bring me to the microscopy screen here again. And now you can see there are a bunch of different microscopes. Okay? And you can see this one, and I'll have to move my screen down just a little bit. This one says TEM. That's the Transmitting Electron Microscope. And you're going to select the thumbnail images here and view them, and they will appear in a screen above. Okay? And you'll be able to see the different organelles here. You can click on labels, and it will tell you which organelles are which. The mitochondria, the plasma membrane, the cell wall, the lipid granule. And so it's going to ask you some questions about those organelles. And the next picture also has that lipid granule there, the nucleus. And finally, the last picture is going to show you the process of asexual reproduction, which is budding in the yeast. And yeast is the only single-celled fungi. Otherwise, all other fungus are made up of multicellular organism. Let's look at the lab sheet then. Kind of see what the next part is. Then it asks you, to look at modal protists. And even though we're calling this the fungus lab, remember that a protist is not a fungus. And an amoeba is not even a fungus-like protist. It's an animal-like protist. So it's not just fungi that you're looking at. It's just that's the very first thing that you'll see in this portfolio. So it says, some unicellular eukaryotes are modal, which means they can actively use mechanical methods to move in specific directions. They usually have flagella, or in the case of the amoeba, they're using their little pseudopods to kind of creep and move like the blob. You will now examine modal species, each which uses a different method to move about. Select amoeba in the species selector. Amoeba move by extending projections of cytoplasm known as pseudopods or pseudopodia. In the compound microscope, watch a video of amoeba moving. Okay, so again, we're going to go back to this thing up here. And I can just go to Protista and go to amoeba. And then I can go down here to the compound microscope. So you'll see some slides here that allow you to look at and identify, how the amoeba moves. And I'll let you watch those and come up with your descriptions. Now you're going to select the paramecium. Paramecium move cilia, or very short hair-like projections, to act somewhat like oars on a rowboat. Use the compound microscope to view a paramecium moving. Then you're going to select euglena. Euglena move using whip-like flagella found at one end of their cells. In the compound microscope, watch the video of euglena swimming. Those would be plant-like protists. Oh. I swear euglena have... thought euglena had chloroplast. They do, yeah. I don't know why they wouldn't be categorized under plant-like protists, though. So photosynthetic protists, it says... Oh, it's talking about euglena. I'm sorry. It says, in addition to capturing and engulfing microorganisms and other sources of food, euglena use chloroplast to harness energy from the sun. These are very unique, euglena, in that they actually both are, they're like photosynthetic heterotrophs. So they can make their own food, but it's not enough to sustain them, so they also eat other things. So they're kind of interesting in that way. And then it says also click on diatom in the species selector. Diatoms are another group of photosynthetic protists. They produce intricately patterned shells made of silicon dioxide, which is basically glass. So they're kind of cool. They're one of my most favorite types of plankton. If you could have a favorite plankton, which I'm sure you all have a favorite plankton, but, it's probably the one who lives in Bikini Bottom and is looking to steal the recipe for the Krusty Krab, Krabby Patty, right? Most diatoms are non-modal. Observe the compound microscope and scanning electron microscope images of diatoms. So again, the same dealio, guys. You're going to go to protists, you're going to look up diatoms, and you're going to look both here at dissecting microscope and compound, they said compound and scanning electron microscope images. I suppose you could always look at the dissecting scope too. Dissecting scopes don't give you very, very magnified images, and they give you kind of the outside view. Whereas usually with a compound microscope, sometimes you're going to be able to see more inside the cell. So if you want to picture kind of just looking at the silicon dioxide shell, the dissecting scope probably is your best bet. Then we have parasites. Some protists are parasitic, not all of them. We're going to be observing 2 Giardia and Trypanosoma. And you're going to do that by selecting those from the species selector. And it says, based on the micrographs and observations from part B, suggest how individuals of these species move around. So remember, we have movement by pseudopoda, we have movements by cilia, we have movements by flagella. So which one of these is, or more than one, is being used by these particular parasites for motion? Now we're going to looking at fungi-like protists. So these are slime modes. And slime modes are oftentimes, they're single-celled, but they live in little colonies. So they're colonial organisms. So they kind of live together even though they're single-celled organisms. And they're like fungi in that they have similar types of life stages. They have different types of reproduction, similar. They have a fruiting body like a fungus does, that kind of thing. So it says, based on the size scale indicated by the micrographs, is a cellular slime mold fruiting body a millimeter, a micrometer, or a nanometer scale structure? So a millimeter would be a thousandth of a meter, a micrometer would be a millionth of a meter, and a nanometer would be a billionth of a meter. So about how big are these guys is what we're saying. So scientists traditionally categorize protists by splitting them into fungus-like, plant-like, and animal-like groups. However, new research includes genome-level molecular analysis has shown that this traditional system does not represent protist diversity accurately. Of the species you observed above, which is most difficult to classify as fungus-like, plant-like, or animal-like? Explain your reasoning. Okay? So, you can't really get this one wrong. You can, I guess. I mean, there's one that's going to be the most difficult to categorize. But I want you to give me a really good explanation as to why one doesn't fit well into one of those three categories. Okay, if it's plant-like, animal-like, or fungus-like, why wouldn't it fit well into either any of those three categories? That's it, guys. It's not a very difficult lab as long as you get that whole online microscope lab thing up and functioning. If you have Make sure you save this, by the way, before you attach it then to the Dropbox. If you have any additional questions or concerns, feel free to give me a call at extension 2204 or send me a webmail message. Have a great day, everyone.